Here is about a bacteriology topic, that is Bacillus, and in this video I am discussing about two important bacilli that are Bacillus Anthrax and Bacillus Cereus. First, about Bacillus Anthrax. It is a causative agent of zoonotic disease anthrax, and we know that it is widely used as a bioweapon. And about its morphology, it is gram-positive, large rectangular rods, and it is arranged in chains, and it is non-motile and capsulated, and it bear non-bulging oval spores. It is the gram staining of the Bacillus anthrax. We can see large rectangular rods, gram-positive rods, arranged in chains. Then about its pathogenicity. Pathogenicity depends upon two virulence factors, that are capsular polypeptide and anthrax toxin. First, about capsular polypeptide. It is a polypeptide capsule, and it is made up of polymer of glutamic acid, and it inhibits the phagocytosis, and the capsular polypeptide is plasmid coated. Then second virulence factor, that is anthrax toxin. The anthrax toxin is composed of three factors: edema factor, protective antigen factor, and lethal factor. The edema factor, which is an adenyl cyclase, which increase the host cyclic AMP level in the cell and cause edema. Then protective antigen factor, it binds to the host cell and facilitate entry of other factors like edema factor and lethal factor. And the third factor, that is lethal factor, which cause cell death. Individually, these factors cannot cause a pathogenic effect, but the whole complex produce local edema and generalized shock. Then clinical manifestation, anthrax. It is a zoonotic disease. Then three clinical forms are present. The first is cutaneous anthrax, second pulmonary anthrax, and third intestinal anthrax. First, about cutaneous anthrax. The cutaneous anthrax is otherwise called Hyde-Porter's disease because it is common in dog workers. Then the entry is through skin, and it is present as malignant pustule. In the figure we can see a malignant pustule. In malignant pustule, a papule is developed at first, then it is developed into a painless vesicle, then it converted into a cold black necrotic ester surrounded by non-pitting induration edema. Then a satellite lesion filled with serum or yellow fluid is present surrounding it. It resolves spontaneously, but fatal septicemia or meningitis in a few are present if it is untreated. Then pulmonary anthrax. It is also called Wool-Sotter's disease because inhalation of dust from infected wood is the cause of pulmonary anthrax, and it clinically presents as hemorrhagic pneumonia with a high fatality rate, and the complications are hemorrhagic meningitis, bacteremia, hemorrhagic mediastinitis. And the third is intestinal anthrax. The injection of spores through the meat is the cause of the intestinal anthrax. It is rare, but violent enteritis with bloody diarrhea is the clinical feature. Then it has a high case of fatality. Then other classification of the anthrax is industrial and non-industrial. The industrial anthrax include hides, hair, wools industries. The non-industrial anthrax include agriculture, exposure to animals. Then about its lab diagnosis. In lab diagnosis, specimen collected are an ear cut off from the animal, swab soaked in blood or blood smear or pus or CSF. In the microscopy, we can see gram-positive large rectangular rod-shaped bacilli, and it occurs as mainly chains. It is the gram stain. We can see gram-positive large rectangular rod-shaped bacilli, and the McFadden's reaction is used for the capsular demonstration. Blood smear are stained with polychrome methylene blue. We can get an amorphous purplish material surrounding the blue bacilli. It is the McFadden's reaction. And immunofluorescent test is used to detect capsule antigen, and it is used as a confirmatory test. Then Ascoli's thermoprecipitation test is also used. It's a ring precipitation test. Then culture. We know the Anthrax bacilli is aerobe, and it can survive temperature range between 12 to 45 degrees Celsius, and the spores appear only in the culture or the soil. Then the spores are central or subterminal oval spores, and the spores are not found in animal tissues. In blood agar, non-hemolytic colonies are get tri-wrinkled non-hemolytic colonies are get, and in nutrient agar, frosted glass appearance is also observed. Then medusa head appearance. Under low power microscopy, the edge of the colonies is composed of long interlacing hairs of bacilli resembling locks of matted hair, and it is called medusa head appearance. It is the medusa head appearance. Then in culture, bacilli arranged end to end long chains. The ends are truncated or concave and swollen. It is called bamboo stick appearance. Then we know it is non-motile. Then another important reaction is string pearl reaction. Solid medium containing 0.05 to 0.5 units of penicillin per ml. In that medium, cells become large, spherical, and occurs in chains or on the surface of the agar in 3 to 6 hours. And the Bacillus anthrax is susceptible to gamma phage. We use selective medium such as PL80 medium, that is polymyxin, lysozyme, EDTA, thallus acetate medium. Then identification. It is catalase positive, and it ferment sugar with acid production. The gelatin liquefaction test, we get inverted filtry appearance. It is the gelatin liquefaction test, we get inverted filtry appearance similar to this. Then animal inoculation. Apply the infected tissue over the shaved skin of guinea pig. It causes fatal infection. The antigen detection in tissue extract is also preferred. Then Ascoli's thermoprecipitation test. Then molecular methods like PCR, AFLP method. It is amplified fragment length polymorphism, and MLVM is also used. It is multiple locus variable number of tandem repeats. It is a full form. Then we know the anthrax spore is widely used as a bioterrorism, and the guidelines for identification of the anthrax bacilli in case of bioterrorism is any gram-positive bacilli which is non-motile and non-hemolytic in blood agar, with the morphology and cultural characteristics similar to those of anthrax bacilli, and it is catalase positive, and initial confirmation is done by lysis of gamma phage and direct fluorescent antibody test for capsule specific staining and for polysaccharide cell wall antigen, and further confirmation is done by PCR. Then about prophylaxis. In prophylaxis, general method like improvement of the factory hygiene, proper sterilization of the animal product, or cases of animal died of anthrax should be buried deep in quicklime or cremated, and active immunization like Pasteur vaccine, strain vaccine. In strain vaccine, spores of non-encapsulated virulent mutant strains are used, and Masuki vaccine. In Masuki vaccine, spores of a stable attenuated carbazo strains in 2% saponin is used. Spore vaccine for animal use only. It is protective for 1 year. Then alum precipitated toxoid vaccine, that is protective antigen is used there. It is for the human use when occupational exposure is possible. 3 doses are given for 0 dose, 6 weeks and 6 months. Then annual booster dose are also given. Then treatment include doxycycline, ciprofloxacin, and also for prophylaxis use. Then antibiotics are not effective in this case. Then about Bacillus cereus. It is an important anthracoid bacilli. It is motile and non-capsulated. We know the anthrax bacilli is non-motile and capsulated. It is just opposite to that. And it is not susceptible to gamma phage. Then it does not react with the anthrax fluorescent antibody conjugate, and it is not pathogenic to animals. It causes food poisoning. Two types of food poisoning is caused by this bacilli: diarrheal type and emetic type. The diarrheal type is caused by serotype 2, 6, 8, 9, 10 and 12. Except 9, all are even numbers. And emetic type, it is caused by serotype 1, 3, 5, odd number serotypes. Then the diarrheal type is caused by diarrheal toxin, and emetic type is caused by emetic toxin. And there is a difference between the incubation period of two type of food poisoning. In case of diarrheal type, the incubation period is 8 to 16 hours because the toxin is secreted in the intestine and which leads to diarrhea. But in case of emetic type, the incubation period is only 1 to 5 hours because preformed toxin causes the emetic type of food poisoning, and it is heat stable. But in case of diarrheal type, the toxin is heat labile. Then meat, vegetable or dried beans, cereals are the food contaminated items which cause diarrheal type. And in case of emetic type, rice, mainly the Chinese fried rice, is the contaminated food through which emetic type of the food poisoning occur. Then selective media like mannitol, egg yolk, phenol, red polymix in agar, that is MYPA, are used. Then treatment is usually this disease is self-limited, no specific treatment is needed. Then it is a multiple choice section. You can done it yourself. Thank you.